Good morning. My name is Caroline Coleman, and today we are going to discuss the basic molecular structure of DNA and RNA. We are going to discuss their differences and similarities, as well as how to construct a DNA and RNA molecule. So, as you can see on the screen, they're already written up some comparisons about their different characteristics and their function and what they do for us in our body. So you can go ahead and write those down as I walk through that information. So with DNA, it contains two long strands wrapped in the format of a double helix structure made up of nucleotides. It is more stable than RNA and stores our genetic information. It determines what gets passed down and is also in charge of what takes place in the cell. So it has a foundation that switches between a deoxyribose sugar and a phosphate group. And we will discuss the deoxyribose sugar and how it compares with RNA in a little bit. We will actually draw those structures out as well and take a look at seeing the differences with those. It has a nitrogen base back up the middle, connecting parts of the double helix, and those are held together by hydrogen bonds. So we have the purines A and G, and those always go with the pyrimidines T and C for DNA. Now RNA, there's a difference, and we will get to that. A single DNA molecule has thousands of genes, and it also has complementary base pairs. So moving on to RNA, it is single-stranded and is a 5-carbon sugar, which is ribose, and it has one more oxygen than deoxyribose does. And we will be able to see that difference when we draw out those two different sugars later on. It instead has the base uracil instead of thymine. There are 3 major types: messenger RNA or mRNA, ribosomal RNA or rRNA, and transfer RNA or tRNA. And its major job is protein synthesis. It converts the information that's held by DNA that allows it to turn into proteins and then be processed by the rRNA. So it basically decodes the stored information from DNA. All right, go ahead and grab some different colored markers or pens or pencil, whatever you want to do. And we're going to go ahead and draw out these structures. I encourage you to draw them out with me, as it will make it easier for you to understand and learn this information and be prepared for your assessment later on this week. All right, bear with me as we start with DNA. It is very difficult to draw on this whiteboard. Oh, goodness. Let's erase. Oh, it erased the whole thing. All right, well, we'll just start over. All right. Okay. There we go. All right, and then we will go ahead and draw those rungs on the ladder in green like I talked about earlier. And so these are those complementary base pairs that are going across. And to reiterate, these are held together by the hydrogen bonds as well. And we talked about that A is going to go with T, and G is then going to go to C. All right, so there's our DNA for us. Now let's transition to RNA. We'll go back to red. This one's a little easier because you just gotta draw one little red squiggly. And then we'll go to green for our base pairs. Whoop. not fully connected, but you get the picture. All right, now, oh, let's see. We have the A is instead going to go to U, and G still goes to C. So that does not change. All right, now let's go ahead and transition to drawing our sugars. And we're gonna number our carbons. You've taken organic chemistry, you know what this is, numbering the carbons. And then down here, we have our OH group. And here, we have our H group. All right, now let's go and draw the same structure for, and that's an oxygen, not a zero, just so you know. All right, so we have these, they look very similar, except they're gonna have that extra oxygen over here like we talked about earlier. So this is where the extra oxygen comes into play right there. All right, so we have these, and now we have these little branching off right here, as you can see. So we can have tri, di, or monophosphate groups. So we'll put those in pink. Those are phosphate. Wow, it is hard to write. There we go. Have a little key. Have the same thing over here. All right, now we're going to switch over to do orange, where we can have our purines or our pyrimidines coming off. So those will come off right here. And let's see, so we can have a pyrimidine, excuse me, let me draw that one first, which looks like that. We're not going to write this whole word because that would be extremely painful for everyone involved. We're going to have the purine, which looks like this. All right, and then those can attach here or here. Either one can go there. All right, so that about wraps up all we are going to talk about today. As always, if you have any information, please let me know and I'd be happy to re-explain it to you. Please study this information as it will be on your assessment at the end of the week. Have a great rest of your day.